Okay, so you just asked what is the deal with our exams. So here's the plan. I want to have, you guys are still gonna have a lecture exam and a lab practical. The lecture exam will still be multiple choice, true, false, and maybe matching if I can make that work online. I need to like find a test maker program that I like and see how much creativity I can figure out for our exams. If I can't, then it'll be multiple choice and true and false. And that's just how it's gonna be. It'll be based on histology and integument. So it'll be what we talked about on Tuesday, all of it, all of chapter 4, and on integument, which is what we're gonna talk about today. Your lab exam, it'll be scheduled for the same day that it's scheduled for on our syllabus, as in that day, which is Thursday. Your lecture exam will open on Thursday. It doesn't necessarily have to be at 5:30, I guess, but it'll open on Thursday, and then you will have 3 or 4 days to access it. And then once you open it, you will have a hour window, damn it, sorry, to finish it. I brought in a house plant that was too wet, and now I feel like I've got like these annoying little flies around. Or maybe it's just one harassing me. Anyway, for the lab exam, same deal with opening it on Thursday and giving you a 3-day window. The difference is to try and keep it as much of like a practical as possible. I'm going to have that one be identifying images. So it'll be as close to the practical as I can get. So I'm thinking 25 images with 2 questions each. Basically like, what are we looking at? Or like name the structure that the arrow is pointing to. Or where is this type of tissue found in the body? Those types of things, but with all questions. And so the lecture exam will be 50 questions and the lab exam will be 50 questions, and they'll be both will be open for like 3 days, and you'll get an hour for the lecture exam and an hour and a half for the lab practical. Does that answer your question?
Yes, thank you, Professor. Appreciate it.
Feel better about a little bit better about that, I hope.
Yeah, thank you.
I haven't started writing either of them yet, so I've yet to find out what kind of a nightmare it is to try and use a like an exam, a test generator program, and there's a lot out there, so I have a lot of research to do to find out which one is the one I like best and can do the most things. But I'm gonna figure it out, and I will let you know as soon as I do. I'll definitely be keeping in touch, so don't worry about that.
What about the study guide? Have you finished that and decided when it's going to be posted?
Yeah, I'll do that first. I'll work on that first. I'll have it for you by tomorrow night. Does that sound okay? Sorry about that. It's supposed to be a week ahead of time. Tomorrow night. I need to write your guys' quiz 5 tonight too. I'm like behind. Okay, that's okay. We have like next week and then spring break to like catch up. Not that we don't have skeletal in between. Woo. Okay. So the integumentary system. We're looking at this, and you'll see by your lab manual that this is, if you read that first paragraph, this is a very tissue-oriented approach. So we're gonna have a lot of familiar friends popping back in, like names of tissue-wise. It gets a little bit heavy at the beginning, and then I feel like it sort of chills out, and then we get to do some like sort of medical stuff like burns and cancer and aging and stuff having to do with the skin. So it begins. If I can, hey, there it is. Okay. All right, so your integument. Again, we are starting chapter 5. This is on page 153 of your textbook, and we're on lab 10 in your lab manual, okay? We're kind of knocking these out at the same time, like we did with histology. So your integument has 3 layers, okay? Your epidermis is the superficial most superficial layer of your skin, the one that you can touch on the outside, right? Epi as in outermost or on top, epi, like epitome, right? epidermis is the most superficial region. Your dermis is the region right directly under that, so it is just deep to the epidermis. This is the layer that is, it's the thickest part of your skin, and it's connective tissue, and you got all of your cool like glands and hair follicles and stuff that lives in the dermis. It's also the layer of skin that ink is deposited into when you get a tattoo, because your epidermis like regenerates, right? It sloughs off because it's that stratified squamous epithelium, if you'll recall. But your dermis stays the same, so they deposit ink. A good tattoo artist can deposit ink like directly just in the dermis without getting anything into the epidermis, or deeper where it can like bleed out and stuff. So in case you're wondering, I learned that from your textbook, which is actually pretty cool. They mentioned that. And then finally, deep to the dermis is the hypodermis. So hypo as in hypodermic, right? So below the skin, hypodermis, otherwise known as superficial fascia. Don't worry about that. I could probably just delete that actually. What you need to know is that it's the deepest layer of the three of your skin. It's actually not technically a part of your skin proper, okay? So the epidermis and the dermis are your skin proper. The hypodermis, the layer directly underneath, is really just like a fat layer, the fat layer that's underneath your skin. So it just kind of just means under the skin, the hypodermis, but it's not technically part of the skin, okay? I know that's a little bit weird, but. Okay, so don't let this freak you out. Just focus for this moment on these three words, these three layers, right? So the epidermis in this picture is this brownish layer here on top. So you can see that it's pretty thin. It's stratified squamous epithelium, which means that it's not vascularized, right? You don't see any blood vessels reaching up into this brown layer. Whereas the dermis, which is this big, thick layer in pink here, this is all connective tissue in here. And then we've got all kinds of fun structures associated with the dermis. And we're going to go through each one of these and what they are and what they do. And the dermis, of course, is highly vascularized and innervated. So you'll see nerves and veins in the dermis. And the hypodermis is right deep to that, and it's mostly fat. So it's mostly adipose tissue, right? Adipose, loose connective tissue, right? As per Tuesday's lecture. Okay, so we're gonna go through these other things as we go along, but the epidermis, dermis, and hypodermis are the three layers of your skin, okay? So let's go through them one at a time. The epidermis, again, this is stratified squamous epithelium. It is keratinized, so it contains the substance keratin, which is strong and almost plasticky, and it can be soft or it can be hard, but it's very waterproof, which is the most important function that it provides to our skin. So it is stratified squamous epithelium, and those cells have keratin around them, okay? Within and or around them. The cells of the epidermis, those stratified squamous epithelial cells, can include also keratinocytes, which are cells that produce keratin, okay? So again, remember the suffix "cyte" tells you that this is a mature cell, and this type of mature cell is found in the epidermis, and it produces keratin, okay? The protein keratin. You also have melanocytes, again, a mature cell that produces melanin, okay? So 10 to 25% of the cells in the lower epidermis, so the deepest part of the epidermis, are melanocytes, and they are producing melanin. Which is a pigment, right, which gives the skin its color. People with darker skin don't necessarily have more melanocytes. Instead, the melanocytes that they have produce more melanin. We all have the same number of melanocytes, roughly. We also have epidermal dendritic cells, which are for immunity or for protection against pathogens in case of injury. So these epidermal dendritic or Langerhans cells are macrophages, which means that they're large and they eat pathogens, okay, through phagocytosis. And we also have tactile touch recepting cells at the very, very, very deepest part of the epidermis, very close to the dermis. So the vast majority of the epidermis is dead, keratinized, stratified squamous cells, okay? So everything that we're looking at here in purple in this picture, in purple, this is all the epidermis, okay? The dermis is this white part down here. So in this particular picture, we are looking at all the different layers of the epidermis, okay? So I'm gonna go back real quick. So we're looking at all the layers of this right here, right? And there are multiple layers, and you do have to know the names of them, but there is a fun way to try to remember. So the layers are from superficial to deep: the Stratum Corneum, the Stratum Granulosum, the Stratum Spinosum, and the Stratum Basale or Basale. This picture is of a piece of thin skin. So from anywhere all over someone's body except for their palms or the soles of their feet. So the palms of your hands and the soles of your feet are thick-skinned, and they actually have one additional layer that isn't seen in this picture, and that is the Stratum Lucidum, and it would usually go right here. So it'd be in between the Stratum Corneum and the Stratum Granulosum. The Stratum Lucidum would be right here. I'll show you a picture of thick skin in a second. So the differences between these different layers are, because this is biology, not like arbitrary, but a little bit fuzzy, right? So the Stratum Corneum is I'd say easiest to identify because it is the largest of the layers of the epidermis. It is the most superficial in addition. These, so we've got the cells of the stratified squamous epithelial cells that are of the stratum corneum of the epidermis, so the superficial most layer, are, they're not alive, right? They're dead. They're basically just sacs of keratin at this point. So everything that you can touch of your skin is dead cells, little sacs full of keratin, and that's it. And that layer is the thickest of the epidermis and is called the stratum corneum. The stratum lucidum, which is only present, again, in thick skin, so the skin of the palms of your hands or the soles of your feet, would appear right here, and it will appear there in another picture that I'm going to show you. It's called the stratum, well, this is the stratum corneum because it's the, they call it like the horny layer, but it's like the hardest, most keratinized layer. The stratum lucidum is so named because it is like kind of transparent, the cells there. So if this was an example of thick skin, the stratum lucidum, which would be right here, would kind of look like a light line near the border between the stratum corneum and the stratum granulosum down here. I'll show you a picture of stratum lucidum in a bit. But the stratum granulosum, which is directly deep to the stratum corneum in thin skin, which covers most of your body, is 3 to 5 layers of flattened cells. The organelles are deteriorating, so they're kind of on their way to being dead, flat sacs of keratin, like in the stratum corneum. The cytoplasm then is full of lipid granules and keratohyalin granules, which is, as you might imagine, keratin, sort of like cartilaginous granules in there. So that's why it kind of looks sort of grainy. You see like all these little dots in there, and why it's called the granulosum, like granules, okay? So the stratum granulosum is just deep to the stratum corneum in thin skin, and just deep to the stratum granulosum is the stratum spinosum, which is also known as the spiny layer, because it's kind of hard to tell here, but these cells have these little like spiky sort of like little arms, so they kind of look spiky, sort of spiny and spiky. So this is the stratum spinosum. You've got lots of keratinocytes. So again, these are cells, adult cells that produce keratin. And they're unified by desmosomes. And desmosomes, again, remember, are the little cell junctions. But they are also, actually, let me just confirm this. I'm getting this wrong. Okay. Is that what I mean when I say that? Yes. Unified by desmosomes. Okay, so those are the cell junctions that are like little tack welds, right? They hold them together really, really strongly. You have also lots of cytoskeletal elements inside of those cells made of pre-keratin. So we're producing keratin. It's not granulated keratin, and it's not just a full-on cell of a sac of keratin yet. At this point, the cells are still alive, and they are starting to produce keratin. And then the deepest layer of the epidermis is the stratum basale. We've got actively mitotic cells in this layer, right? So this is where the skin actually grows from. So the cells that are produced here will push, as they're produced, push the cells above them, superficial to them, up and away towards the surface of your skin, right? So the surface of your skin is constantly sloughing away because the cells in the stratum basale are constantly dividing and pushing the cells up and away. So so the newly formed cells become part of the more superficial layers. And then it also contains, this layer contains melanocytes and epidermal dendritic cells, which basically move elements around like keratin and melanin. Okay, so let me see what's next here real quick. Ooh, we're going into each one. That's fine. Any questions before I go into detail? I'm just gonna go into detail, and then I'll ask if you have questions after that.
I just had a quick question about the actual lab. So I'm like looking at it as we go through. Yeah. But is this lab going to be due at the same time as 6 through 9?
I think it's due on the 7th, the integument lab on Canvas. Okay. I'll check that. I believe that it is due on the 7th.
Okay.
Lab 10 due April 7. And that's just because the way that I wrote my syllabus was a little bit stupid, and I had the lab for integument happening after the exam. I don't know what I was thinking. Or no, it wasn't that. It wasn't the syllabus, was it? It is. This on the syllabus. So stupid. So I moved it up, and I figured that you guys would understand, since we want to just get it out of the way. We're going to do integument today, and then we're going to get tested on it on Thursday and not have to ever look at it again, right? Otherwise, I'd have to put the integument lab stuff in with the skeleton lab exam 3, which would be weird. So I figured we get tested on histology and integument all at the same time and be done with it.
Okay.
Yeah? Okay.
Thank you.
Yep, no problem. Okay, so we're starting down in the deepest layer, so that's stratum basale all the way down here. And we're going to work our way up, going more and more superficial. Okay, so the deepest epidermal layer is, again, the basal layer or the stratum basale. It is the deepest epidermal layer. It is attached to the dermis, right? So it's just superficial to the dermis, which is the layer underneath the epidermis. It is a single row of stem cells, right? So these are the stem cells of your skin. They are actively dividing, and then they differentiate into either skin cells that are going to end up moving more superficially and become the cells of the stratum corneum eventually. Or they may become melanocytes or dendritic cells, helper cells. This is also called the stratum germinatum. Don't worry about that. But that's just because there's lots of germination happening, right? Lots and lots of rapid division. The journey, woo, pardon me. Hey, calm down. The journey from the basal layer to the surface, to the stratum corneum, takes 25 to 45 days. So that's how long it takes for new cells to basically reach the surface of your skin. Kind of cool. Okay, so once again, that layer is here. So just superficial to that, we have the stratum spinosum. And this is, again, these are going to be cells that have a, we've got pre-keratin filaments, so we're starting to produce keratin. They are attached by desmosomes, which again are those tight junctions like tack welds that hold cells really, really strongly together. We also have lots of melanin granules and dendritic cells. So again, dendritic cells are cells that are moving melanin granules around or moving keratin granules around in and amongst the other skin cells. So kind of like dispersing them around in the skin. And it is also known as the prickly layer because the web-like pre-keratin filaments and stuff sort of make them look sort of spiky, which is kind of weird. So from here to here, we've got the stratum spinosum, and they're looking a little bit spiky here. Okay, just superficial to the stratum spinosum is the stratum granulosum. So that is going to be here. And it is relatively thin, 3 to 5 cell layers thick. The cells are starting to flatten, they're starting to die. The organelles are starting to deteriorate in these cells, which is how it says right here. And so the cytoplasm is full of granules of like pre-keratin. This is, again, a keratohyaline. So hyaline is in like hyaline cartilage, so sort of like cartilaginous little granules of keratin. And again, it is these granules present in the cells at this time that caused somebody to name this layer the stratum granulosum. And finally, the superficial most layer of the epidermis is the, here we go. Okay, so sorry. So the stratum granulosum is here. And then since this is an example of thin skin, we don't have that here, but there is another layer in between the stratum granulosum and the stratum corneum, and that is the stratum lucidum. And the stratum lucidum is, again, only in thick skin, which is on your palms and the soles of your feet. It is, it's thin and it's kind of transparent, so it looks a little bit lighter in color in stained slides or samples like we are looking at for this class. They are flat and dead keratinocytes. So they have lots and lots of, they produce a lot of keratin, and so there's a lot of keratin around and within them. So that's why the thick skin of your hands and the soles of your feet is a little bit tougher, right? It keeps them nice and tough because they are subject to the most like friction, and we really don't want to disrupt like the layers of your epidermis, right? So that additional layer just gives it a little bit more thickness and toughness. So here's a picture that is comparing a sample of thin skin to a sample of thick skin, where you can see the stratum corneum in both. But then in the thick skin sample, you can see the Stratum Lucidum, which again is this sort of like white, sort of ghostly looking layer right above the Stratum Granulosum, which is this dark line here. So again, on the thin skin, that is not present. You don't have like a sort of a whitish looking strip right there. The Stratum Granulosum, that very thin layer is here. The Stratum Spinosum, here to here, and the Stratum Basale is here on this thin skin sample. And then the dermis, right, which is connective tissue, is just deep to that. On the thick skin example, we've got a really clear delineation between the Stratum Basale all through here and the dermis, the connective tissue of the dermis, right? So let's see what kinds of, oh, that's not what I wanted. I wanted that and go to Lee's. Go to Lee's. Let's see what we got for a skin sample. Cool. Okay. So here we are in Lee's Histology virtual histology lab. I'm going to zoom way in to this. This is the apical layer of this tissue, right? This epithelial tissue. It also is the most superficial layer of your entire body, right? So this is your skin. Let's see. Simple. Okay, so we're just looking at the epithelium here. That's fine. If I, oh, this is all kind of dead here. But what you can clearly see from this picture is the line between the underlying connective tissue of the dermis and the, so the dermis, as you can see, is very, very large, contains lots of blood vessels and other structures. And then deep to that, you've got the hypodermis, which is the fat layer, right? So you've got hypodermis here, dermis all through here. And then the epidermis is relatively very thin. The stratum corneum is the thickest part, so it's all this dead stuff here. And then you've got your other layers here, which are really hard to differentiate in this picture, and I would not ask you to tell me what layer of the epidermis you're looking at in this picture, besides the stratum corneum, which is the dead outermost part. Let's see. Maybe in connective tissue. What do you have to show me here? Oh, awesome. Okay, so we've got, yep, stratum corneum indicated right here on this sample. We've got, just deep to that, the stratum granulosum, right? So how cool. It is actually showing us where those layers are. So here's the corneum, again, the outermost dead cells layer. The granulosum, where you've got lots of granules of keratin and keratohyalin present in there. Deep to that, the stratum spinosum, which is from here to here. So I guess you'd have the granulosum around here. The spinosum is this area that looks a little bit more purple versus the red of the layers superficial and deep to it. And finally, the stratum basale, which they are indicating is right here. So this darkest layer of cells, just superficial to the connective tissue that makes up the dermis. So that's pretty cool that we get to see all of that. That was in the histology in the connective tissue version of the skin sample in Lee's histology. Based on this picture, based on what we know about these layers, corneum, granulosum, spinosum, and basale, can anybody tell me if this is a sample of thin skin or thick skin?
Thin.
It'll be thin skin because we are missing that stratum lucidum layer. Yeah. So the stratum lucidum layer would be here-ish, but it's not. So this must be a sample of thin skin right here. Let's see what we got. So the integument thing in the Wiley Plus, I'm not even really sure like what is useful about this, honestly, because they don't even really do anything besides take off the they take off the epidermis and the dermis so that you can see the hypodermis. So at in this level of dissection on Wiley Plus, this is the hypodermis that you're looking at. And that's basically it. It's like, oh, well, there's the epidermis and the dermis, and you take those away and you got the hypodermis. So not terribly useful. Ooh, that's lovely. But luckily, we have our whole histology collection, which includes skin samples. So let's see if we can find one of those. Section of skin. Yes, don't mind if I do. All right, what do we got here? Dead cornified layer of the epidermis at the surface, pulling away from the living cell layers beneath it. Yes, I did it again. Okay, good for me. All right, so again, the stratum corneum is the dead stratified squamous epithelial cells, but they're dead sacs full of keratin really at this point. Are we gonna be shown the actual layers, or is it gonna be? Oh, awesome, cool. Okay, so stratum granulosum, stratum spinosum, and let me guess, stratum basale. Excellent. So this is a really cool one too. Again, is this an example of thin or thick skin?
Thin.
Thin again, because we don't have stratum lucidum. Let's see if we can find stratum lucidum in one of these other ones. Maybe, maybe not. Okay, well, maybe not. All right, so I may not have a good picture of stratum lucidum for you besides the one in the actual PowerPoint, which is here. So I would have to be clear either that, hey, stratum lucidum is here, is this thin or thick skin, I might ask you, or I would say, this is an example of thick skin, please identify this layer. Okay, hope that sounds fair to you guys. And any questions about the layers of the epidermis at this point? No? Then I'm gonna move on. Hey, is that Josephine? Mute your mic. Somebody mute their thing. There, that's better. Okay. Nope, still there, still going. Oh well, I'm gonna keep going. All right, so, oh yeah, to wrap up, I didn't really do my whole big page on the stratum corneum. So we did our stratum lucidum. We see it here. And then stratum corneum, again, it's the thickest layer, 20 to 30 rows, dead, flat, keratinized, membranous sacs. Okay, so they once were cells, but are no more. Again, thickest part of the epidermis, 3/4, 75%. It functions as a protective barrier from abrasion and penetration, right? This is important. I'm gonna keep things from getting into the internal, to the inside of your body. It is waterproof, or it waterproofs as a verb, I guess. And it's also functions as a barrier against biological, chemical, and physical assault. So there are things on your in your stratum corneum that actually allow your skin to fight against biological pathogens, against chemical harm, and as well as like physical abrasion and stuff like that. Okay, so wrapping up that. Oh, here's a cartoon of the same thing. So we got another description of all these layers. Once again, not including lucidum, it looks like. And in this picture, you can see a melanocyte, a cartoon melanocyte here. So again, this is a cell that produces melanin. We've got one of those epidermal dendritic cells that I keep mentioning. So this guy is basically going to be distributing those melanin granules that are produced by the melanocyte throughout the rest of the epidermis. And here is a little sensory nerve that does kind of extend up into the epidermis from the dermis. So here's my easy, fun way of, that I stole from another website, full disclosure, of remembering the layers of the epidermis. Starting with the name, losing the stratum part, but starting with the actual name. So we go corneum, lucidum, granulosum, spinosum, basale. Come, let's get sunburnt, which is ironic since we're going to talk about cancer as a result of too much sun exposure, right? Okay, come, let's get sunburnt. And so that includes lucidum, which is only present in thick skin, right? Come, let's get sunburnt, going from the superficial most layer to deep, superficial to deep. Come, let's get sunburnt, okay? So hopefully that'll help you guys out a little bit. And I will move on to the dermis. Let's see where we're at. We're doing good, okay. All right, so the dermis is just deep to the epidermis, right? So we have all of those different strata of the epidermis, and just below the stratum basale is the dermis. So we've got lots of strong, flexible connective tissue in the dermis. So the cells of that connective tissue include fibroblasts, which again are cells that produce the fibers that are found in connective tissues, right? Macrophages, which again are immune cells that eat pathogens through autophagy or phagocytosis. And occasionally mast cells and white blood cells, which are also immune cells, immune-related cells. So these are really very important, those last three, because the dermis is subject to injury. We want to make sure that we've got lots of immune power, like right there. So it's literally right under the surface of the epidermis, right? Right under the surface of the skin. You're ready to fight anything that might try and get through. There are 2 layers of the dermis. Luckily, it's not 5 or 6 layers like the epidermis. There's only the 2: your papillary layer and your reticular layer. So again, don't panic. We're just going to focus over here on the dermis, which we're talking about now, which is, again, this big, thick layer of connective tissue with all of these fun appendages associated with it. And here are the 2 layers of the dermis. The papillary layer is really, it's the layer where you've got these bumps here. So you can see how it's kind of wavy all along here at the border between the dermis and the epidermis. That waviness, those dermal papillae, which cause that waviness, that whole region, like from here to here, is the papillary layer of the dermis, okay? Everything below that of the dermis is the reticular layer, okay? Yeah. Okay, so let's talk about the papillary layer a little bit more. This is areolar connective tissue. So there is collagen fibers associated with that, as well as elastic fibers and lots of blood vessels. So the dermal papillae, which are those bumps that make up the papillary layer, contain capillary loops, Meissner's corpuscles, which are a type of sensing nerve ending, and free nerve endings, which are another kind of sensory nerve that we're going to go through. So the papillary layer is where you find these dermal papillae, and within the dermal papillae you will find these capillary loops, basically, right? So highly oxygenated blood reaching basically like the end of the road in your circulatory system, right? And making that bend, going around the bend to try to go back to the heart. So now you've got oxygenated blood reaching all of those cells of the epidermis, right? All of those stratum basale cells that need those nutrients to divide to produce more skin or heal your skin, dropping off that oxygen becoming deoxygenated blood, indicated by this blue color here, and traveling back all the way back to the heart to be reoxygenated again. And then you've also got these free nerve endings in the dermal papillae, which are responsible for certain types of touch sensations. And the Meissner corpuscles that are mentioned on that slide are specific types of sensory cells. They look a little bit like the Pacinian corpuscle that's down deeper, but of course, since they're only in the papillary layer, they would be found like up here in the papillae, and there isn't one. There is not one in this picture, but it would be up here, and it would be like a little, kind of like a little balloon, kind of like this little guy. Okay, and again, they are a sensory nerve that detect certain types of sensations. All right, and then the reticular layer of the dermis is, again, all of the rest of it besides that papillary layer. It makes up 80% of the thickness of the entire dermis. Still areolar loose connective tissue. So we've got collagen fibers to provide strength and resiliency, right? So we like to have that collagen. We like to maintain or add collagen to our face in particular, right? It keeps you looking young. It's that strength and resiliency that collagen fibers provide, and elastic fibers for that stretch recoil, right? So that you can kind of still got a little bit of that. Still got it. All right. The epidermal ridges are basically the result of the papillary ridges in the papillary layer of the dermis, okay? So your fingerprints is, the technical term for your fingerprints is epidermal ridges, okay? They lie on top of those, the papillary ridges, and they form these ridges that you can actually see on the surface of your skin. So the ridges themselves, I'm sorry, I'm rearranging again. Here we go. The ridges themselves have sweat gland ducts that open out onto them. And that's why whenever you touch something, you end up leaving a fingerprint, because those little sweat ducts are constantly producing sweat. And so that gets deposited along the ridges of your fingerprints. So that's pretty cool, right? So the ridges, these epidermal ridges, they give you not just a very unique individual identification system, but they also provide greater surface area on your fingertips for sensing things. So your fingertips are very, very sensitive, right? And that's because you've got all this extra surface area, and all of those sensory nerves associated with the dermis are all very highly concentrated on your fingertips. And that extra ridges in the surface area that it gives also makes them kind of grippy. So it actually helps us to be able to grip things too, which is kind of cool. And the study of your fingerprints is called dermatoglyphics. I just thought that was cool. That's just an FYI, kind of interesting kind of thing. You don't have to remember that. I just thought it was kind of cool. Hey, every time I do that. Okay. All right. So in addition to those epidermal ridges, there are also such things as cleavage lines. And your book goes into not a terribly large amount of detail, but it's interesting. It's on page 160, a little clinical connection box there. I thought it was really cool though, because the collagen fibers of your dermis are actually arranged, those bundles are actually arranged like in a pattern throughout your body. So that pattern is what is called cleavage or tension lines. And those cleavage lines kind of fall on the lines that are indicated by this image here, which is kind of crazy, right? It looks like crazy tribal tattooing or something, but this is actually the lines in which the collagen of your dermis naturally lies. So whenever you do surgery, if you care about the cosmetic appearance of the scar, or even the healing ability of the scar, you make your incision based on these cleavage lines. So I thought that was like pretty fascinating. And I always wondered why my little appendectomy scar was like in this like very specific sort of diagonal plane, and now I know why. So pretty cool, right? I thought so. Okay, so let's talk about skin color. Are there any questions about any of that? Try and pause before moving on. I got somebody on the chat. We'll be covering integumentary for lecture as well. Okay, since we're not going to have a transcript of the chat, since I'm just doing YouTube videos, I'm just going to answer audibly. Oh, maybe I'll type it in. So yes, if I can. Yes, we're going to be doing, we're going to basically be doing the same thing as we did on Tuesday. We'll cover all of chapter 5 during lab and lecture Zoom sections. Time left over, we'll use for answering questions for Q&A. All right, so hopefully that answers that for you guys. I still got time. I did set an alarm. Ooh, hello. I did set an alarm for 5 o'clock so that I won't go over, so I can move over to the lecture. I don't even know if this is gonna be, I'm wondering if this is even a good pattern to maintain. Like, should I just leave one session open the whole time maybe? And we can take a break when we feel like it. And that way I'll just like be on for 4 hours, you know, with a break depending, for you guys to come and go and ask questions and whatever.
I have a quick question, Professor.
Okay.
As far as the layers of the dermis, the papillary layer, I know you went over the dermal papillae, what it contains, the capillary loops, the Meissners. Yeah, I'm sorry. We're going to need to know what each of those do, right?
Yep, and I'm going to go through them. Yep, we're going to go through the various structures of the dermis shortly. Yeah.
Oh, okay. So you just kind of briefly just went over what they were.
Yeah. Yeah.
Okay, and then one more.
In detail.
Got it. And then one more question. In the dermis layer, obviously, you said that there are fibroblasts, macrophages, and mast cells and white blood cells. We're going to need to know those 4 different things are required to be a part of the dermis, right? Do we need to know exactly what they do? Does that make sense? I'm sorry.
Yeah, no, I believe that they are just part of the, oh yeah, so those are part of that areolar tissue of the dermis. And if I'm not mistaken, I think they are common, I think that's just like areolar tissue. I think that's just an areolar tissue thing. Yeah, so actually, if you go over areolar connective tissue, loose connective tissue, those are the types of cells that are associated with it. So it's not weird or specific to dermal areolar tissue. It's areolar tissue in general has macrophages, the fiber-producing fibroblast cells, and the mast cells.
So like you said, they're not just specific to the dermis. They're in each layer of the skin, even the dead skin cells.
Think about the different tissue types. So go back to Tuesday momentarily, as painful as it might be. The epidermis is epithelial tissue, right? So closely packed cells, right? It's stratified squamous epithelium specifically. So it is the little flat pancake ones in multiple layers, right? That's the epidermis, which includes the stratum corneum, granulosum, lucidum in thick skin, spinosum, and basale, right? You still with me?
There you are. I'm sorry, Professor. I think my internet cut you out.
Oh.
Yes, go ahead.
Are you okay with what I just said?
Yeah, I'm sorry.
Okay, no problem. So the epidermis is the epithelial tissue. The dermis, just deep to that, is made of connective tissue. the type of connective tissue that it is made of is areolar loose connective tissue specifically. It also happens to have a bunch of other stuff in it, like various nerve endings, hair follicles, sweat glands, and stuff that interact with the surface of your skin. But the areolar loose connective tissue that the dermis is made out of still stands with its structure as described in chapter 4. So on page 133 of your text, where it describes areolar connective tissue, it says that it is made up of collagen fibers, and that the cells inside of it, as the loose areolar connective tissue, include the macrophages, the mast cells, and the fibroblasts. Does that make sense?
Got it. Okay, I see what you're saying. When you explained it that way, where it's the simple squamous tissue, which is the epidermis, and then connective tissue, which is the dermis.
Exactly.
Got it. And so any connective tissue is gonna have these fibroblast macrophages. Am I understanding that correctly?
So the dermis does, because it's areolar loose connective tissue.
Got it.
Specifically has those cells associated with it.
Got it. So any other part of the body, if it's connective tissue, it's going to have this.
So if it's the areolar connective tissue. So the areolar connective tissue it's the subcutaneous layer. So it's the dermis. It also is a part of mucous membranes around blood vessels, nerves, and body organs. So it's like around it forms a little layer around certain things like blood vessels and organs and nerves, and then it also happens to make up the dermis as well.
Got it.
Connective tissue.
Awesome. Thank you, Professor.
No problem. So like I said, this lecture and your textbook actually, and the lab manual are taking a very like histological approach to the integument, which I think is really cool, because since we just talked about histology and different tissue types, now we're getting to see a couple of those different tissue types in detail and like as they're functioning, what they do. So I think that's pretty cool, actually. So we're still talking about I guess we're not. So this was the order that your text took. Talking about skin color and hair before moving into other structures of the dermis. So we just got done talking about the dermis. We talked about the papillary layer at the very top and how it forms ridges. We talked about the reticular layer underneath and how it's made of loose areolar connective tissue. The color of people's skin, animal skin, really, human skin specifically, is, melanin it's a thing with all animals, but some of the other things are not. Okay, sorry. So, comes down to 3 different aspects, or 3 different contributing factors. The first one, of course, is melanin. So remember, I know we just stopped talking about the dermis, but the melanocytes, right? Those were associated with the stratum basale of the epidermis, remember? So in this picture over here, we were looking at the epidermis and all this epithelial tissue. These melanocytes are technically associated with the epidermis, right? So not with the dermis. So these guys are producing the melanin. They are in the stratum basale. The dendritic cells are going to help to spread that melanin through the rest of the epidermis, right? So we're not talking about melanin being in the dermis. It's not. We're talking about the epidermis. So those melanocytes, since they're present in the epidermis, produce melanin into the epidermis. And then that melanin that's produced moves to these keratin-producing cells, the keratinocytes, and together they form what are called pigment shields. for the nuclei of all the cells that they are nearby to. So the important part, or the important function of melanin really, is to absorb UV radiation, okay? So melanin actually helps you to fight against UV radiation. So the lighter your skin is, the more prone you are to burning, and therefore the more prone you are to your DNA being affected by UV radiation. So that's why you have to practice greater protection from the sun, right? Wear your sunscreen. But we'll talk more about that when we get to skin cancer. And then freckles and moles and stuff like that are basically just little tiny areas where melanin is has accumulated, is concentrated in the skin, and they're not like a problem. It's not a problem. It just is a thing that some people have. All right, so that's one factor that affects skin color. The other two are keratin and hemoglobin. So keratin is a yellow to orange pigment. You may remember from biology, from Bio 1. We looked at keratin pigment a little bit. This pigment in our skin is most obvious in the palms of our hands and the soles of our feet. And it is a precursor to vitamin A. So it actually requires that the sun to produce vitamin A. And the hemoglobin is, of course, the protein in your blood that carries oxygen. And that blood, that red oxygen-rich blood, is what gives the pinkish hue to your skin. So the three different factors that affect skin color are melanin, keratin, and hemoglobin. Any questions on that? Make sense? Okay. All right, so we've got, now we're going back into the dermis, okay? So those, the melanocytes, the keratin producing or fixing cells, and well, your hemoglobin is gonna be your dermis, right? Because your blood doesn't actually reach into your epidermis, right? Because it's epithelial tissue, which is avascular. But going back down into your dermis, going down, diving down deeper now again, let's talk about some of those all those crazy accessory structures that are found in the dermis of the skin. So within that loose areolar tissue, which is made up of those mast cells, macrophages, fibroblasts, in an extracellular matrix of collagen and elastic fibers, right, as loose areolar connective tissue, there are also these other things that are associated with the surface of your body, with your skin. So let's talk about hair. Hair is actually important, and all mammals have it all over their bodies, right? Even whales and dolphins have a fine layer of fuzz on the surface of their skin. It alerts the body to the presence of bugs and creepy crawlies on your skin. So I don't know if you've ever noticed this, but if you I don't normally shave my arms. My sister shaves her arms, but I never really have. But the one time that I did, I tried it once. It freaked me out because it felt like my arms were numb. And I feel like literally when like things would land on you, like it would be numb. You couldn't feel it. And when my arm hair finally grew back, which I'm glad it did because it keeps me warm. It helped me, like, you can actually feel things like crawling on you, which is like kind of crazy. So you don't have that ability without your hair being there. So it actually helps you to like sense touch on your skin, believe it or not. As far as why humans have long hair on their scalps, I think it's still kind of up in the air as like an evolutionary question, but prevailing thoughts are that it guards your scalp against physical trauma. So it actually adds a little bit of a layer of protection to your scalp, which is like full of blood vessels and obviously is protecting your brain and stuff. So also protects against heat loss. So it keeps you warm because heat rises. So if it rises out the top of your head, your hair tries to help to keep that in. And it guards your scalp against again, UV radiation. So it keeps your scalp from getting burned because it is the part of your body that's usually closest to the sun or directly facing the sun. So the entire surface of your body has hair, has at least a fine layer of hair, except for the palms of your hands, the soles of your feet, your lips, your nipples, and certain parts of your external genitalia. So parts that like do not necessarily need that kind of protection, or parts in which that kind of protection would actually like interfere with you trying to like do certain things, right? So generally speaking, thick skin is hairless skin, and thin skin is hairy skin, okay? So generally speaking, thick skin does not have hair, and thin skin usually does, except for those exceptions. So your hair, like your nails, consists, and like the surface, like the stratum corneum of your skin, of your epidermis, consists of dead keratinized cells. So again, these are cells that are not alive and they are full of keratin. They contain hard keratin. So unlike your skin, unlike your epidermis, where the keratin is like kind of soft, the keratin of your hair and nails is relatively hard. So it's more durable, can take more of a beating than the soft keratin of your skin. There are pigments associated with hair follicles. So again, we've got melanins, there are melanocytes at the bottom of the root of hair follicles, and they can produce colors ranging from yellow to black. When your hair turns gray, it's because the melanin has been cut off, the production has been decreased, and there's an increase in air bubbles in your hair shaft. I actually didn't know that. That's kind of weird. All right. Okay, so this little pink line right here is your epidermis, okay? This underneath here, all of this really, I suppose, is meant to be your dermis, okay? So this guy here is one of your hairs, and it goes all the way down to a bulb. Everything that is below the skin surface is considered to be the root, and everything above the surface of the skin, visible to the outside, is considered the hair shaft. Your hairs are usually associated with a sebaceous gland, which is an oil-producing gland that we'll talk about shortly. In order to keep that hair waterproof and soft, you need those oils. And also associated with every hair in your dermis is one little tiny muscle called an erector pili muscle. And that one little muscle, when it contracts, pulls your hair upright. So that's basically how you get goosebumps. So every little goosebump that you get on your skin when you're cold or when you're scared even is because all of your little erector pili muscles contracted and pulled the hair upright. So that helps to trap warmth against your body. And at one point when we were more furry, it's thought that it helps to ward off predators by making you look bigger. So like when cats get really big and puffy when they're scared, it makes them look bigger for all of their hair to stand up on end. So that's just kind of like a leftover, an evolutionary leftover from when we were furry. But it does keep us a little bit warm, I suppose. And then this right here is a cross section of a hair follicle. So you've got a cross section, as you can see, this little plane, this little transverse plane here reflects what we're looking at here. We've got the, oh, I've got to rearrange again, go away. Okay, we've got the actual hair right here, okay? So the, it does consist of a cuticle on the outside, a cortex in just deep to that, and the medulla in the middle. And then it's surrounded by the follicle, which includes a connective tissue sheath and several different membranes. which I'm not going to ask you to memorize. What I want you to know is that the hair shaft is above the skin, the hair root is below, the base of the hair is the hair bulb. The bulb and the root live in a hair follicle, and every hair has an associated sebaceous gland, usually, and an erector pili muscle, okay? So you don't have to know like all these different layers of the follicle wall and everything, just the different parts of the hair. I hope that sounds good to you guys. Oh, and here is an actual picture with all of that same stuff. So this is the actual hair, and the rest of all of this is the follicle that it's living in. So down at the bulb down here, you've got melanocytes and blood vessels and things that support hair growth. So keratinocytes, right? Cells that produce keratin are going to be here as well, because that's how your hair grows, right? It grows from the bottom out. I keep doing that. Okay, so the hair follicle again extends from the epidermal surface down into the dermis. Not worried about the layers of the hair follicle, but that it terminates, the deepest part of your hair is called the root, or called the hair bulb, okay? The hair bulb is where the hair grows from. You've got melanocytes and blood vessels and keratinocytes associated with that part of the hair follicle where the hair bulb is, so that the hair can grow. And from there, you've got the root. And then when we reach the surface of the skin, everything outside of your body that you can see from the outside is the hair shaft, okay? Oh. Don't worry about the root hair plexus, but it does stimulate the erector pili muscle, which I do want you to know. So this is your little muscles. It's actually made up of smooth muscle attached to each follicle, and when they contract, you get goosebumps. It pulls your hair up, right? Okay. Really just worried about these structures here. So just focus on this picture here. This obviously is not associated with this plane. This instead is a zoomed in version of this, which would be more of a longitudinal section up and down. And again, I don't need you to know the different layers of the hair itself or of the follicle. Just know these terms here and what they are and what they do. Okay. And another one, they're just full of pictures here. Okay. Okay. There are two different types of hair, generally speaking. Vellus hair is the pale, fine body hair of children and adult females. And terminal hairs are coarse and long. These are the hairs of your scalp, of your eyebrows, your axillary, so like your armpits and your pubic regions, and the face and neck of males, right? And me sometimes, I get chin hairs, I'm not gonna lie. You're not alone, it's a thing. All right, and then hair growth. This is more like for your curiosity, in case you care. Yeah, whatever. That's cool, but ain't got time for that. Hair thinning and baldness. Alopecia is hair thinning, can happen in males or females after the age of 40. There are also people that have alopecia from the time they're born. I knew a guy that didn't have any eyebrows or eyelashes or any body hair. It happens. And then true baldness that we normally think of and that we keep trying to treat with Rogaine and stuff is actually genetically determined, so you can't really treat it. It is sex influenced, so it happens more often in males, and it's caused by a follicular response to whatever this hormone is that I can't think of right now. Nope, can't think of it. But it's genetically determined. usually happens in males, and it's when your follicles respond to a certain hormone after a certain age, usually. Okay, other structures in the dermis. Let's talk about those sebaceous glands, many of which are associated with hair follicles, again, so those oils help to keep your hair soft and waterproof. But they are widely distributed over the entire body's surface. So they're not just associated with hair follicles, they can actually open directly out onto the skin without a hair follicle. But most of them develop from hair follicles. They become active at puberty, of course, right? So you start getting acne and problems around that time, because it's when those sebaceous glands like first become activated. Sebum is the word for the oil that sebaceous glands secrete. Remember we talked about the different types of exocrine glands. Sebaceous glands are holocrine glands. So let me remind myself what the heck that means. I should probably remember, but I don't. Glands. Okay. Holocrine. Secretion contains large amount of lipids and a plasma membrane. So holocrine was the one where the entire cell on the inside of that duct or that exocrine gland ruptures, and then that sebum, secretion from that gland includes both the actual secretion itself and all of the parts of the cell that exploded and went in with it. The sebum of sebaceous glands is bactericidal, which is kind of cool, right? So that's part of the biological protective barrier that your skin provides. So those glands, those exocrine glands, open up onto the skin surface, sebum reaches the skin surface, and then it actually helps protect us against certain forms of bacteria. And then again, it softens the hair and the skin, so it keeps our hair and skin from drying out. Okay, so here's that picture again. Did I do it? I did. So here's our sebaceous gland. This is what we're focusing on right now, right? So here's another sebaceous gland that is associated with a hair follicle, but again, it doesn't necessarily have to be. It's just that most of them are. And here is an actual image of a sebaceous gland with the duct here at the top, which would be more superficial, right? Be closer to the surface. We've got secretory cells down here. So these are the guys that are actually producing the oil. And here is our hair root, right? In the hair follicle. So this is a sebaceous gland that is associated with the hair follicle. Cool. I don't want to forget that I've got all this stuff and that we are talking about, that's not what I want. We are talking about, we're all supposed to be in lab, right? We're supposed to be in lab right now. So let's make sure that we are looking at lab resources here. I thought I saw a hair cross section here somewhere. Through hair bulb, follicle, thyroid gland. I'm curious about, sweat gland. That's cool. Here's another hair follicle. Mammary gland, submandibular, sebaceous gland. Hello. What have we here? Okay. Section through the dermis that cuts through a sebaceous gland and the collagen of the dermal layer. And that's as zoomed out as we get. And that's, okay, that's cool. Okay, so I think what we're looking at here, I think we've got the hair follicle over here. This must all be a sebaceous gland here. These would be our secretory cells. And up here somewhere, up here somewhere would be the duct, right? Assuming that this is not upside down. which I suppose it could be. An apocrine gland, we're gonna get there. Section of pigmented skin, I didn't see that, that's cool. Erector pili muscle and sebaceous gland, neat, neat, neat. Okay, so this must be the erector pili muscle. This looks very much like smooth muscle here, right? It's not, so they're sort of long and skinny cells, each one with a nucleus. And then this is going to be that sebaceous gland. So we've got our secretory cells all in here. And then I'm assuming that the duct is going to be, the superficial area is going to be up here. Through the dermis, cuts through sebaceous gland and the collagen of the dermal layer. So I guess we're able to see some collagen fibers as well. To the left of the gland is the thin slip of erector pili muscle. Note the slender spindle-shaped smooth muscle fiber cells. Cool. Yes, I have noted that. Thank you. I hope you guys have noted it as well. Ding dong, someone's joining us. Welcome.
Okay.
All right. And pigmented skin, 100 times of the entire integument from the epidermis to the subcutaneous layer of the hypodermis. So we've got our adipose tissue here. Looks like we've got our connective tissue here. That's the mailman, and the dogs are going crazy. Sorry about that. Oh, never mind. Sorry. So yeah, adipose down here, connective tissue, and we've got all the layers of the epidermis here. Oh, I'm so sorry about the barking, you guys. We've got UPS, not for me. UPS for the neighbor. Must be nice. I want a package. All right. So the stratum basale of the epidermis, right? Stratum granulosum, spinosum, corneum here at the top, the dead stratified stuff here at the top. So we already went through that, so I'm not going to torture you with that anymore. Let's instead move on to sweat glands, okay? So this is another structure that's found in the dermis. This is, again, in that loose areolar tissue that makes up the dermis layer of the skin. Oh God. I don't know if you guys can hear this, but it's ridiculous. All right, there are two main types of sweat glands, also known as sudoriferous glands. Sudoriferous glands, okay? That's just a fancy word for a sweat gland. There are eccrine sweat glands, which are merocrine glands. So again, those are the ones that those are the ones that just secrete their product without the cells breaking down. Okay, so holocrine ones are the ones that break down. Yep, holocrine ones are the ones that break, the cell actually explodes and the entire cell and the product, the secretory product, go out the duct of that exocrine gland. Merocrine glands are the opposite end of the spectrum, so they're the ones that just secrete by exocytosis, their secretion. So sweat glands, one type of sweat gland are eccrine glands. These guys are super abundant, especially on the palms of your hands, the soles of your feet, and your forehead. So of course, these guys are going to be producing sweat, which contains a lot of stuff besides water and salt. Kind of interesting. And so the ducts connect to pores on the surface of the skin. Of course, a pore is just a tiny hole in your skin, right? So of course, every duct is going to terminate superficially at a pore, which is just the opening. Eccrine glands, eccrine sweat glands, function in thermal regulation. There are sweat glands that don't. We're going to talk about that. But these ones, the eccrine sweat glands, are specifically the ones that function in thermal regulation, right? So they help to cool you down when you get too hot. That's either because of physical activity or even like emotional sweating. In both of those cases, eccrine glands may be activated. So here is an example of an eccrine sweat gland in our pretty picture. So here's the pore, right? Which is the terminus of the duct, right, of the gland. And that's the gland itself right here. So if you were to take a cross section of a piece of skin of a sweat gland, you wouldn't really be able to see like the outside of it. That would be not two-dimensional, right? This is roughly two-dimensional. And of course, you end up cutting through sections of this coiled tube, which is what an eccrine gland is in essence. So you end up having like some transverse, some longitudinal-ish cross sections of different parts of this coiled tube. So all of these are different parts of the eccrine sweat gland. And if you look closely, it looks like we have, ooh, even there, look at that. That looks like, actually looks like stratified or simple cuboidal. Which is not surprising since those things are often found in glands, as are a lot of other epithelial tissues, as we found out on Tuesday. So eccrine sweat glands function in thermoregulation. They look like a coiled tube with a duct that terminates in a pore on the surface of your skin. Apocrine sweat glands are apocrine. They're actually not. They're not apocrine glands. Your textbook, I think they're just going by the most current information, and that most current information apparently is that the sweat glands that are actually named apocrine do not function like apocrine, like normal apocrine glands. So I don't need you to know this. I just think it's interesting. It's and kind of cool actually that the text makes sure that it uses the most relevant and accurate recent information. So they actually secrete via exocytosis. So it's actually more of a, they're more like eccrine glands, but they still call them apocrine glands, apocrine sweat glands, whatever. Apocrine sweat glands produce not so much sweat as more of like an oily secretion. You can find apocrine sweat glands in your axillary, so like your armpits and your pubic region, right? So these guys all become activated again at puberty because all that fun stuff happens at puberty, right? You end up with your sebaceous glands being activated. You also end up with your apocrine sweat glands being activated, and they, because they secrete a sebum that is sweat plus fatty substances and proteins, which doesn't actually have an odor itself, but once it reaches the surface of your skin and interacts with bacteria that is living normally on your skin, like good bacteria, then it produces body odor. So that's why you start to have body odor when you reach puberty as well. So the reason for it even existing, besides just being annoying and selling Axe body wash, is that they might actually be sexual scent glands for humans, which is a really interesting theory or hypothesis, I guess, technically. But I don't think we really know for sure. And yeah, so maybe study reproductive biology, you know, be the one to find that out. All right, there are also some other specialized apocrine glands, and they include the glands that are inside of your ear canal that produce earwax. They're called ceruminous glands, and mammary glands, which of course are in females, which produce milk, right? The mammary glands of the breasts. So that was two different types of sweat glands we talked about: eccrine sweat glands and apocrine sweat glands, of which there is not a specific picture of here. Nor do we have one, I think. Let's find out real quick. I know we had a really cool, oh, we do, look at that. An apocrine gland section through the coiled secretory portion of an apocrine gland. Simple cuboidal epithelial cells. Yeah, check that out. So that's pretty cool. So we got a nice picture of an apocrine sweat gland where you can see, at least down here, it's really nice and clear, a single row of nuclei indicating simple cuboidal epithelium, right? That's pretty nifty. Very cool. So this looks like the lumen of the gland. So if this is a cross section, then this might actually be like looking down the tube of the duct, if that makes sense. So that's cool. We have a sebaceous gland. Oh, we did a sebaceous gland. We want to see a eccrine sweat gland. Coils of sweat gland. Awesome. Okay, cool. So this looks a lot like the picture in the PowerPoint, but again, this is just a section through the coiled part of an eccrine sweat gland. So again, this is like functioning in thermal regulation. This is just producing sweat only. And again, we've got simple cuboidal epithelium, right? Forming the walls of the duct of this gland, right? So that's pretty nifty. Very cool. Yep, looks a lot like this, right? Okay. All right, eccrine sweat glands and apocrine sweat glands. Finally, your nails, okay? So your nails, again, made out of hard keratin, the hardest keratin of all, it ends up being in your nail. They're actually a modification of your epidermis, so they come directly from your epidermal layer. On the distal, right, the distal most and dorsal surface of your fingers and your toes. So this is tightly packed, hard, dead keratin. And there are a couple of different associated terms that you'll need to know. The body of the nail, of course, is like the largest surface of the nail. The free edge of the nail is the part that is no longer associated with your fingertip, right? So some of us like to grow those super long. I like to chop mine off because I always get dirt and crap under there. I hate it. The nail bed is the surface underneath the body of the nail, okay? So the nail bed is soft epidermis tissue. The root of the nail is the part that is not seen down past the eponychium. The eponychium is a fancy word for your cuticle, okay? Eponychium is your cuticle, and the hyponychium is where the nail attaches to the nail bed at the distal tip of your finger, okay? So the eponychium is your cuticle, and the hyponychium underneath is where your fingernail attaches to the nail bed at the very tip of your finger, okay? Those are the terms that I want you guys will need to know. The luneal is also a good one. It's the that little white part that you see down at the base of your nail. But the body of the nail, the free edge of the nail, the eponychium and the hyponychium and the nail bed are the important parts of fingernails that I want you to know. Okay. How are we doing? Okay. Your book then goes into, I feel like it's kind of like jumping around, but whatever, types of skin. And I mentioned this already. Generally speaking, 2 types of skin. The first is thin or hairy skin. So covering most of your body, the skin is thin and it contains hair follicles. And then you have thick skin. So the skin of the palms of your hands and the soles of your feet is thick skin, and it's also hairless skin. Thick skin has an extra layer of the epidermis, right? The stratum lucidum that is not present in thin skin, okay? And it is thick because of the stratum lucidum, but also because the stratum corneum, which is the most superficial layer, has many additional layers of dead keratinized cells. So that's the other reason why it's so thick, right? It needs to be good and thick because it's going to be subject to the most abrasion, right? The bottoms of our feet when we're walking and the palms of our hands while we're interacting with our environment. Okay, functions of the integumentary system. This looks a little bit different than what is in your book, just in terms of the order of these, okay? They're not in order of importance or anything. All of the same functions are present in this PowerPoint as in your book, so don't worry about that. Let's talk about protection. We already have 3 types of barriers. We've talked about this. The physical mechanical barrier, of course, against abrasion, right? So we've got protection against abrasion and protection against things actually physically like coming into your body from the outside. The keratin and the lipids present in your skin layers block water and water-soluble substances from getting in through your skin. Certain lipid-soluble substances can be blocked as well. So that's a physical barrier. We're talking about like large molecules, I guess, but I would kind of group that in with chemical barriers, which is basically what your skin actually does to protect against chemical barriers specifically, is that you actually have secretions. The secretions on your skin are slightly acidic, so that helps to kill bacteria. That acidic environment on your skin, slightly acidic, kind of cool. And then you've also got the dendritic cells and the macrophages associated with the dermis, right, and the epidermis, the dendritic cells, which are part of your immune system. So they function to protect against pathogens. Just know the 3 different ones, right? Chemical, biological, and physical barriers. This is gonna really bug me if I don't fix this. Oh, why you gotta do me like that? Okay, I have to fix it now. I'm gonna be really annoyed. Okay. All right, carrying on. Okay, chemical, biological, and physical layers are all part of the protection function of your integumentary system. Additional functions include body temperature regulation, right? So we perspire, right? So our eccrine sweat glands produce water and salt and a bunch of other things, and we call that fluid sweat. And as it evaporates from the surface of our skin, it takes some of our body heat with us, so that's good when we get too warm. We also have the vessels of the dermis, the blood vessels of the dermis will also dilate if we get too warm, and so the blood that is in the core of our body that eventually reaches the surface of our skin to those capillaries of the dermis. If those capillaries are wider, we can get more blood to the surface, and so that heat, if it can reach the surface of our body, it can escape more easily. Also, cutaneous sensations, super important, right? Our skin, our integumentary system lets us feel and interact with our external environment. Depending on the type of sensory receptor in the skin, which I don't think I included the different types of sensory receptors in the skin. Then you don't have to know them. You don't have to know the different types, just know that they exist. That there are nerve sensory receptors in your skin for touch. The different types of them, because I didn't go over them, I don't expect you to know the different types, but depending on how close to the epidermis they are within the dermis, like the Meissner corpuscles that we talked about, you don't have to know the name of them because I didn't go over it. I don't have them in the PowerPoint, I forgot. They're very close to the epidermis, so they're better for very, very fine sensations. And then those free nerve endings that we talked about, again, you don't have to know what they're called. They're just a sensory receptor for you guys, but they help you to feel temperature, right? So you can feel whether whatever you're touching is hot or cold. And then, of course, pain receptors. So those free nerve endings also contribute to pain reception. the Pacinian corpuscles that were down deeper, the deep part of the dermis. Those guys help you to feel like super like bassy vibrations, like deep sensations. So it depends on, you know, where they are in the layer of the skin and how sensitive they are. And there's different ones that do different things. But you don't have to know the different names of them or what they do. Just know that there are sensory receptors and that they are, for the most part, located in the dermis layer of your skin, okay? Your integumentary system also functions in your metabolism, right? So you also synthesize vitamin D in your skin, or at least the precursor is produced in your skin with the help of UV light. Collagenase, since it's an ACE, right? We're talking about an enzyme which breaks down collagen. So your skin does help you to produce certain important molecules, and it actually serves as a blood reservoir. So up to 5% of your body's blood volume at any given time is in your skin. It's actually part of your integumentary system. And excretion. Believe it or not, when you are sweating, part of that sweat includes nitrogenous wastes, which includes things like ammonia and urea, and salts. So you actually do excrete a certain amount of, I don't want to say like toxins, but waste products and possibly toxins too. So yeah, so those are the 6 functions of the skin that you need to know: thermal regulation, protection, blood reservoir, sensation, excretion, and metabolic function, namely vitamin D synthesis. Any questions about that? Okay, moving on. I'm going to brush, no? I'm going to brush past wound healing we're not going into as much deep detail as we possibly could on this. There are 2 main types, and that's because your textbook really doesn't go into super detail on this, at least not with like visual aids. So this is what we're gonna do. There's 2 different types. There's epidermal wound healing. This picture is actually on page 169 of your textbook. And then there is deep wound healing. Okay, so there's wounds that affect only the epidermis, and then wounds that go deeper than the epidermis, extending down into the dermis, right? So in epidermal or shallow wound healing, it happens a little bit deeper, or a little bit different, excuse me, because your epidermis, being an epithelial tissue, is avascular, right? There are no blood vessels in your epidermis, okay? So if you only scratch your epidermis, which is usually like in the form of like an abrasion, you just kind of scrape, you know, some of those surface cells away. What happens is that the cells of the stratum basale are actually going to start migrating in towards the center of the deepest part of the wound. They actually move, so they actually physically move towards each other. And then when they reach each other, there is actually a function of them all like being surrounded by cells again that tells them to stop, which is really cool, and it has a name. I don't need you to know it, I just thought it was pretty cool. Contact inhibition. So once they have contact with cells all around them, they stop moving. So these guys would move towards each other, and then as they produce cells, they are actually going to fill in the gap, right? So to remind you, we've got, of course, we've got the basement membrane for the epithelial tissue of the epidermis, right? That stratified squamous epithelium. We've got the stratum basale, which now has formed a contiguous line again after having been separated here by this injury. The other layers of the epidermis superficial to that, and the dermis deep to the epidermis, right? So these guys migrate in, and then they will produce cells to fill in the gap, to re-thicken that epidermis. So that's if you only scratch your epidermis, right? You didn't get deep enough to get to the dermis. But if you do, if you have a deep wound, this looks like some sort of puncture wound, you know, this is awful, right? So you have some sort of puncture wound that goes through the epidermis down into the dermis, right? And it even ended up severing some sort of a blood vessel here. Yeah, so it damaged the blood vessel, whatever this was. So I guess this could be like the pin from our physical processes lab, right? So when you did that horrible little like blood sample clicky thing, you made a wound like this. So you got the epidermis penetrated, the dermis is penetrated, and even a blood vessel is penetrated here. So what happens is that since the blood vessel was penetrated, whether or not a blood vessel is penetrated or not, you go into an inflammatory phase. The inflammatory phase is when the surrounding blood vessels and capillaries, they actually, the walls, that simple squamous epithelium, right, that single layer, the cells kind of separate enough so that like blood cells and white blood cells and other important things like platelets and stuff from the blood can seep out. They actually want it to leak out. In this case, they don't have to leak out because you already have a hole here. So they just kind of like are on a free-for-all, leaking out of this damaged blood vessel, both red blood cells, platelets, and white blood cells, like macrophages and neutrophils. Those are just types of immune white blood cells.
Professor, quick question. I'm sorry.
No, go for it.
Did you say that the damaged blood cells are what is actually leaking out of the wound itself?
So if you get down, if you actually wound yourself and get down to the dermis, chances are you're going to damage a blood vessel, because the dermis is highly vascularized, right? If you don't get all the way down to the dermis, if you only reach the epidermis, then you probably won't draw blood. But if you get down to the dermis, you're probably going to damage a blood vessel, because there's so many there. And in that case, the blood cells and the white blood cells are just going to like come out of the damage, the opening, the cut that you made. But
got it. So it's more so the damaged blood vessel than the damaged blood cells. Is that make sense?
The blood cells aren't damaged. They're coming out of the damaged blood vessel. Mm-hmm. Exactly.
Got it. Thank you.
Yeah, no problem. Okay, so those blood cells, white and red, escape out of the damaged blood vessel, reaching the site of the injury, and then they're going to, depending on what their job is, they're going to activate like blood clot formation, or they're going to eat the pathogens, whatever could have gotten in there, get rid of debris, that kind of thing. All of that is the first stage of wound healing for deep wounds. And it's called the inflammatory phase. Okay, so that just includes white blood cells reaching the area and starting the healing process. Okay, white blood cells getting to the area of damage and starting the healing process. There are a few steps in between here, but I'm not worried about it. I just want you guys to know that the inflammatory phase starts, and then at the very end, you end up with a maturation phase. So again, your little epithelial stratum basale are going to migrate across the wound opening. This connective tissue is going to be rebuilt with the help of these white and red blood cells that have escaped out of here. These guys, question?
Yeah, really quick. I'm sorry.
No
so is this the same as like burns, like 1st-degree burn, 2nd-degree burn? Is that what it's referring to?
Good question. We're going to talk about burns more. The healing process is basically the same, but with a burn, obviously, we're talking about usually like a larger like area of the body, and it can be shallow or deep. So generally speaking, like if it's a shallow burn, then yeah, you're going to end up with an epidermal wound healing process. And then deeper burns, depending, will be a deep wound healing process. Yeah, starting with an inflammatory phase and ending with a maturation phase, assuming that there hasn't been so much damage that the epidermis is gone, in which case it can't heal all the way.
And the degree just basically means the layer itself, like the first layer, the second layer.
Exactly. We're going to go over that in a minute. Yeah.
Got it. Thank you, Professor.
No problem. No problem. So
just an example, sorry, just as like an example, would like deep wound healing be considered like something extreme, like being stabbed, like a stab wound?
Technically, yes, because it does go through the epidermis into the dermis, even though it extends deeper than that. At like your dermis, your integumentary system is going to try and heal, and it's it's going to try and repair the damage, even if it's gone farther deeper in. So yeah, technically it'll be a deep wound healing, but this includes things as minor as that pinprick. Anything that gets below the epidermis, anything that's deep to the epidermis, into the dermis, whether it stops there or not, is considered a deep wound.
Okay, and the epidermal wound would be considered like a paper cut?
Only if it doesn't draw blood, right? Because
okay.
the epidermis is not vascularized. So any kind of a scrape that you get where you don't bleed is probably an epidermal wound. Yeah.
Okay.
It's super on the surface. Yeah. No problem. So in the end, after the inflammatory phase and a bunch of other stuff happens, really, in order to repair this connective tissue, the blood vessel itself, and the deeper layers of the epidermis, eventually all of this clotting, all the blood clot that we formed here with the platelets and stuff that came out of the blood vessel, will be pushed up and away until it's no longer needed, right? And then it'll fall off. So the scab comes off after like everything is all like completely healed, which is pretty cool. But until then, it kind of like plugs the hole until your epidermis can repair itself here at the surface level. Because really that's the priority, right? The priority is to close the opening. Like we really, really do not want stuff getting in through an opening in your skin, right? So it's really important that your epidermis is able to seal that gap, right?
Now, when it seals, Professor, is that basically the process of the dermis just kind of regenerating out the bottom and then pushing it back out through that wound?
Yeah, so it's going to do the same thing as it does with the epidermal wound healing, where the stratum basale, which are the actively dividing cells, right? These are the mitotically active cells, are going to migrate in as best they can towards each other. So they're going to migrate across the gap. And then once they have closed that gap, just the basement, just that basal membrane, then as they divide, those new cells that they create will slowly fill this space that is being taken up by the blood clot, right? Until eventually all that's left is the scab on the surface. And then
Got it, which is just a bunch of dead cells anyway.
Yep, exactly. Yeah.
Okay.
All those cells that are produced by that stratum basale, they can turn into melanocytes and dendritic cells, which you need as well, right? But most of them will just end up going along into the pattern of the different strata, going up through Spinosum, Granulosum, Lucidum, Corneum, until they're dead sacks of keratin.
Awesome. Thank you, Professor.
No problem. Okay, so we're doing actually really good on time. I'm really excited about this. So let's talk about problems, right, with your integumentary system in particular. So we're going to talk about just a little bit briefly about skin cancer. Most skin tumors are benign, which means that they do not break off and go into your bloodstream or your lymphatic system and reach another part of your body and then start dividing over there. So when a tumor metastasizes, it's because, well, first of all, a cell becomes cancerous because it's screwed up during the mitotic phase, right? So when we talked about mitosis or the cell cycle, we got interphase with G1, S, and G2. Between each of those phases, there are they call them like stops where it's basically like a checkpoint where the cell has to stop and then it checks itself to make sure that everything is still going the way that it's supposed to be going before letting it go into the next phase. So there's like a check between S phase and G2, and then it allows it to go into G2 if everything looks good. And then there's a check that allows it to go into mitosis from G2, right? So cancer usually is caused by screw ups in the genetic code that codes for those checks, right? So if the checks don't happen and the mitotic process or the cell cycle process screws up and goes out of control, spins out of control, then you end up with cancer. So the genetics of cancer are largely caused by certain mutations in what are called oncogenes, or cancer-causing genes, which basically are genes that are, they do a important thing, but if they have a certain mutation, they stop doing their job, and then you're at a higher risk for cancer. So genetics and a family history are risk factors for cancer, and overexposure to UV radiation in particular is a risk factor for skin cancer specifically. But that being said, most skin tumors that happen at all are benign, which means that the cancer starts and it grows and you end up with like a weird like little lump or something, a little mole. But that doesn't necessarily mean that a part of it is going to break off and get into your bloodstream and then end up in another place, which is what happens when cancer metastasizes, and why cancer ends up killing people. So let's talk about skin cancer types specifically. There are 3 major types of skin cancer. There of malignant skin cancer. There is.
I have a quick question. I'm so sorry.
No, go for it.
As far as genetics for skin cancer, can you reiterate that? Why is it genetically? Because obviously it's something that happens in mitosis. Mm-hmm. I mean, is it something in the chromosomes itself that it's like a trait that you carry on? Is that why it's genetically?
Everything that your body does, including directing mitosis, directing the cell cycle, your genes code for that. They code for every little thing that your body does. So there is actually a gene, there's multiple genes in your genetic code, in the DNA in every single one of your cells, that codes for specifically the proteins whose job it is to make those checkpoints in mitosis. They're like little cops, they're like little crossing guards. And that's their job. And they're supposed to be at the cell when it's trying to go through mitosis and supposed to say, okay, stop, we just finished S phase. Let's take a look and make sure everything's cool before I let you proceed into G2. Or before I let you proceed into prophase or metaphase or anaphase. All of those checkpoints are critical for mitosis to happen normally. So when the gene for those proteins that are the crossing guards for the cops, when that gene gets messed up, and then you have no more cops and crossing guards, then you have anarchy, basically, right? So that mitosis, that cell cycle, can go out of control because there wasn't anybody there to check to make sure that it was doing it right. And it only takes one cell for that to happen in, right?
So it's more so like it's not like random assortment. Like it doesn't just, this isn't something that just randomly can happen.
It's something that's happening in mitosis particularly. It could be, so that the genes that code for the crossing guard, the little checkpoint proteins, that gene, any gene in your DNA is subject to mutation. And that mutation would normally happen during like crossing over, like genetic recombination. So it's not so much when one cell divides into 2 cells, that the cell, so basically mutations would be like, remember in S phase, that's when the DNA in your cell that's about to replicate, that's when the DNA replicates first, right? So the DNA replicates first in S phase, and then it goes into G2, and then it goes into prophase, and then you start to see the nuclear envelope divides and the cells condense into chromosomes, blah, blah, blah. when that DNA is first replicated, if by accident your proteins that are meant to be putting the correct, remember the bases, the different bases, nucleotide bases that make up the structure of your DNA. Remember, it has to be like A to T and G to C, T to A and C to G. If when you're replicating the DNA, you accidentally screw up and put a different base where it was supposed to be, a lot of times it's not a big deal. Sometimes it's just an invisible mutation. Sometimes it does something weird, but it's not harmful. Sometimes it does something good, which is how evolution happens and adaptation happens. But sometimes it's something bad. And if it happens to happen in one of those genes that's coding for a crossing guard that's supposed to be doing those checkpoints for your cells, and you don't have a crossing guard for that particular cell anymore because that cell now has that mutation, when that mutated cell divides, when that cell with the mutated DNA divides, and it has no stopping guards anymore, that mitotic cycle it's out of control basically, or it could go out of control. Just because it doesn't have the stopping guards doesn't mean it won't still be okay. But if it does screw up and it doesn't have a crossing guard to tell it to stop, it might go crazy, and that's cancer. That's how that happens.
Right. So basically, it's those stopping guard genes that you're carrying the mutated gene from your parent, basically.
Yep.
Got it. Okay. Thank you.
Yeah. It's fascinating. It's really fascinating and scary because, I mean, that's why cancer is so hard to treat, right? Because it's your own cells. There's no way to really like tell your body. There isn't a really easy way to tell your body to attack only certain ones of its cells when all it sees is self and non-self, right? Those cancer cells they still hold your DNA, unfortunately. So that's why cancer is so hard to treat. So it's really, really trippy stuff, really cool, interesting stuff. Any other questions about cancer in general? All right, so skin cancer in particular, there's 3 different types. We've got basal cell carcinoma, which is the least malignant, so the least dangerous, really. It's the most common. It's highly treatable. I thought I had, I guess I'll just go through this. In between, we've got squamous cell carcinoma, squamous cell carcinoma. This is the second most common and a little bit more dangerous than basal cell carcinoma. And finally, melanoma, which is the most dangerous type of skin cancer. So let's go through them individually. So basal cell carcinoma is when that stratum basale, so that's the deepest layer of the epidermis, right? Those cells proliferate. So a little screw up happened in one of those cells. We lost a crossing guard, right, in one of those cells, and it starts to proliferate too much. It slowly invades the dermis. So it slowly, it grows so fast that instead of the new cells get pushed up, but it also ends up pushing down because it's growing too fast. So it goes down, it invades the dermis and the hypodermis, so it gets down into the fat layer. It happens slowly, which is good, right? Because that way, as soon as you notice it on the surface of your skin, in which it probably just looks like a raised reddish area, a small raised reddish area, if you go in and have it surgically removed, then you're good. It's gone. So it doesn't metastasize, it doesn't go into your bloodstream. They stay tight together, and it grows slowly. So you usually notice it pretty quickly, and you can get it removed, and then you're good. Squamous cell carcinoma specifically has to do with the keratinocytes of the stratum spinosum. So again, this is that prickly layer. It's the one just superficial to the stratum basale. We've got those keratin producing cells, the keratinocytes. Those guys are starting to screw up. So you end up with a kind of a weird, it's going to end up being like a keratinized sort of raised patch. So instead of it being like a little raised bulb, like basal cell carcinoma, squamous cell carcinoma is going to be sort of a rough patch on the surface of the skin. Most common on the scalp, ears, lower lip, and on the hands. And it can be treated by surgery. So if it's small enough, you can excise it and close the hole, which is fine. Or you can treat it by radiation. So radiation therapy seems counterintuitive, right? Because radiation causes cancer, but at the same time, the damage that it causes will affect cancerous cells more and faster than it will affect the surrounding healthy tissue. So if you are able to, and what they try to do with radiation therapy for cancer is try to really pinpoint exactly where the radiation goes, just at the cancerous area. And that radiation will actually cause the cells to become more and more damaged until they basically commit cell suicide, which is, the term is apoptosis, when cells basically like self-destruct. So yeah, good prognosis for squamous cell carcinoma, a little bit scarier than basal cell carcinoma. And then the scary one is melanoma, which, as you might suspect, has to do with the melanocytes. So these are the melanin producing cells of the deep layers of the epidermis. It is highly metastatic. So that means that it is kind of like loose where the cancerous cells, so it always starts with one cell, right? So one cell screws up, it gets a mutation in the DNA that it got from the division previous, and then it ends up screwing up in mitosis and it starts dividing out of control, and every daughter cell that it produces has that same mutation, right? So they're all screwed up. So that's why you end up with cancer. This is specifically when it happens in a melanocyte. Those cells that it produces are prone to breaking off of the tumor into the bloodstream. And when that happens, that little cancerous blob of cells that broke off can end up anywhere in your body, and it'll lodge there, and then they'll keep dividing. So that's when you end up with a tumor in your lung, you know, or your pancreas or something horrible like that. So you have to be really, really vigilant for these guys. You can treat it by excising it, but you'd have to do a really wide excision for something like this. So like this guy would probably lose his toe at this point, or at least half of his toe. You'd have to take out a lot of that over there. And then you'd have to do radiation as well, or chemotherapy, even though it is highly resistant to chemotherapy, which is another reason why it's so dangerous. Oh, immunotherapy, that's cool. So this is relatively new cancer treatment technology, right? They can design certain therapeutic drugs for you that are able to identify cancer cells in the body. I'm not exactly sure how. They have to take your DNA and do it. It's very specific to the individual person. I should look into it though, because it's super interesting, and it's been around for a few years now. So this is becoming more and more common and more affordable for insurance companies and stuff. So that's cool. So you can treat melanoma with immunotherapy. When you're keeping an eye out for melanoma, you can use the ABCD rule, which is super convenient, right? So the characteristics of a melanoma, if you're worried about something on your body and you're worried that it's melanoma, check the A, B, C, and D. A, is it asymmetrical? So like this one here is clearly it's not very symmetrical, right? So it has it's not perfectly round. So things like moles and freckles and other things that are not cancer that are caused by accumulations of melanin, right? They tend to be perfectly round, right? Freckles and moles tend to be perfectly round. If it's not round, that's your first clue, first red flag. If the border is uneven or has indentations, so like this has a very sort of jagged, bumpy border or edge to the actual melanoma, that's your second red flag. If the color is weird or inconsistent, right? if the color is like unusual for other moles or things on your body, or if like you've got like different colors in the same weird things, you've got like some red and black and other weird colors in there, big red flag. And then finally, if it grows, well, if it's growing, then you should probably get it looked at anyway. But if it gets past a diameter of 6 millimeters, which is roughly the size of a pencil eraser, then that's your last red flag. That's D, right? So A, B, C, D, asymmetry, border, color, diameter. Asymmetry, border, color, diameter, to look for melanoma. Okay, next we're going to talk about burns. Any last questions about skin cancer before we talk about burns? Okay, so burns can be caused by heat, electricity, radiation, and certain chemicals, right? We learned that things with high pH can burn you and things with low pH can burn you, right? So strong bases and strong acids cause chemical burns. Burns are characterized by, of course, generally tissue damage, and then also denatured proteins, right? So remember, under high temperatures or in a too high or too low pH, those primary, secondary, tertiary, and quaternary structures of your proteins, right? Remember, they're the order of the amino acids, and then they fold, and they fold again. All of that structure of the protein can break down, and they can unfold and disintegrate. And we call that denaturing. That can happen with a burn. And then, of course, cell death. So you'll have cells dying. Yeah, of course. The most dangerous part of a burn, and the reason why burns are so serious, not just because you're exposing potentially the inside of your body to the outside world, is usually dehydration, actually. So somebody who's been burned was more likely to die fastest from dehydration. And electrolyte imbalance, right? So having the salts that you need to for your body to function properly, and then the fluids, of course, water, that homeostasis will be disrupted, and that will actually kill you because it'll shut down your kidneys. So renal shutdown is your kidneys failing and going into circulatory shock. So your blood pressure basically going to nothing. In order to determine how severe a burn is, or to estimate the volume of fluid loss caused by a particular burn, you can use the rule of nines. So on any given, or on a human being, generally speaking, the anterior portion of your head, so your face, makes up 4.5% of your body surface. The anterior part of your trunk is 18% of your body surface. The posterior of your head is 4.5%. Posterior trunk is 18%. Front of your upper limbs, 4.5. Back of your upper limbs, 4.5. Front of your lower limbs, 9%. Back of your lower limbs, 9% each per limb, right? So 9 for your right, for the front, the anterior right leg, 9 for the anterior left leg, 9 for the posterior right leg, 9 for the posterior left leg, right? So once you add all of that together, you get 100% of your body area, okay? Of your, the surface area of your body. So depending on how much, on like one last little percent is your perineum, which is literally like the little strip of skin between either your vaginal opening and your anus, or between your testicles and your anus. So they count that as your last little 1% to make a full 100%, which I thought was kind of funny. So if you look at a burn patient and you see that they've got like half of their face, and the entirety of their right arm has been burned, then you can say, okay, well, that's 4.5 and 9, so that's 13.5% of their body was burned. So when you like hear in the news about burn, I don't know, horrible things, or on the true crime things we're always watching, people that get burned, right? They say, oh yeah, they were burned on 70% of their body, right? That's where that's kind of where they get this, right? So in that case, it'd be like she was burned on 13.5% of her body, or 14% of her body. Does that make sense to everybody?
Yeah.
Okay, and like depending on what that percentage is, that corresponds to the amount of fluid lost. So once you get past a certain level, then we're talking about a critical burn. Let me see, percentage proportions of body surface area, estimating surface area affected by a burn. I'm wondering if there is. I thought there was a like a critical level. I guess it was over here. Okay, so let's talk about different degrees of burns. I think this is the critical thing that I was talking about. Don't worry, we'll get to that creepy picture in a second. So we've got, generally speaking, partial thickness burns and full thickness burns. Partial thickness burns include both first degree and second degree burns. First degree burns are burns that only affect the epidermis, right? So it's just the epidermal layer, that non-vascularized stratified squamous epithelium, right? And that's when you see sometimes just redness, some swelling and pain. It's a second degree, so this would be a first degree burn up here, sorry. So so like redness, swelling, and pain for a first degree burn when you're only affecting the epidermis. When you get down into the dermis, you're talking about a second degree burn. So the epidermis and the dermis are both damaged, and that's when you end up with blisters. Okay, so that's basically like fluid collecting in between your dermis and the hypodermis, really. Fluid, you're basically, the skin is separating from the hypodermis, which obviously is a very, very bad thing. And then finally, third degree burns, which are the one example of full thickness burns, is when the entire thickness of the skin is damaged. So we're talking about the epidermis, the dermis, and the hypodermis. So what you'll end up having either like full on like charring to ash. It could be cherry red, like cooked meat, sorry, or black, right? Just charred. So you're not going to actually have pain in this instance in the actual burn site, because all of the nerves that were in the dermis have been destroyed, right? Because that's where all of your nerves of your skin are, is in the dermis. So if the dermis is gone, the nerves are gone, or the nerve endings are destroyed. And if the layers of skin, if the remaining skin edges are too far apart, then there's no way for that to heal. The dermis can't heal itself really anyway without the epidermis being there to help it for those stratum basale layers, or stratum basale layer, those cells to reach each other. There needs to be a dermis, and there isn't one in third degree burns. So it can't heal. So you need to do skin grafting usually in order to save somebody with a third degree burn. So yeah, pretty trippy, right? Any questions on burn types?
You said the second degree burn is between the layer of the dermis and the hypodermis. Mm-hmm. So that's when it goes that deep down, right?
Yep, exactly. Yeah. So it goes as deep as the deepest part of the dermis, right? So that would be like the bottom of that reticular layer of the dermis. Right. But not going as deep as the hypodermis, because then that would be a third degree burn, right?
Got it.
So if the epidermis and the dermis are affected, that's when you end up with blistering. You end up with a little bit with that separation. But yeah, to go down deeper into the hypodermis makes it a third degree burn.
Thank you.
Yeah. Yeah. Oh, here we go. Here's the severity of burn thing I was trying to think of. Okay, so burns are considered critical if more than 25% of the body area, as per like your rule of 9, your body surface, has 2nd degree burns, or if more than 10% of the body has 3rd degree burns. If face, hands, or feet have 3rd degree burns, it's automatically critical, right? So that's only 25% with a 2nd degree burn. So that would only be like 2nd degree burns. I mean, that would super suck. I don't mean only, like it wouldn't be a big deal. It would super suck. Would be like if it was just the anterior and the posterior trunk, like if just your trunk was burned. 2nd degree burns, critical. If it's more than 10%, it's 3rd degree burns. If you get a 3rd degree burn, like all up the front of your leg, you know, and part of the back of your leg, critical. And then of course, any 3rd degree burns on your face, hands, or feet, automatically critical. Any questions? Cool stuff, right? I know you guys are like probably more interested in like the medical application kind of stuff. So this was pretty interesting. All right, and finally, we're going to get into some developmental aspects. I'm just going to, yeah, we're cool. We're really good on time. So I'm going to wrap this up, and then I'm just going to hang out until 5 and then close it. And if you guys have questions, cool. And if not, then I'll just be here, you know, no big deal. So to wrap up, developmental aspects of skin. I didn't get a chance to get to this in the histology lecture because it was so damn long and we just didn't get there. This is the last slide of the histology PowerPoint, which you may or may not have seen if you were looking at it yourself. But all this is showing you is basically like, this is a very, very early 16-day-old embryo. It's literally just like a little blob. It's a little blob with this weird like cleavage furrow in here. This is the dorsal surface. So at this point, the cells of the embryo have differentiated into 3 different types. And from those 3 different types, you will get all the different tissue types that your body will end up having, that the body will end up having. So it is already determined which surface is its dorsal surface and which surface is its ventral surface, which is pretty cool. The predetermined differentiated dorsal surface cells are called the ectoderm, and that's in blue in this picture. The ventral surface, all the cells that are on that ventral surface are going to end up being endoderm and everything that endoderm ends up producing. And all the cells that are in between, sandwiched in between the ectoderm and the endoderm, are defined as mesoderm, and the mesoderm is going to end up being muscle and connective tissue, right? Because we talked about mesenchyme. Mesenchyme is basically like the next step after mesoderm. So mesoderm is like super basic. It's like, okay, all the stuff in the middle. Ectoderm is all the stuff on the outside, and endoderm is all the stuff on the inside. So we're talking about like the donut, right? So this is like the outside of the donut. This is the inside of the donut. Sorry, this is the whole, inside the whole of the donut, right? The exterior surface of the whole of the donut. So lining the epithelial, right? Or the, the digestive tract, starting to lose it. And then the mesoderm is all of your muscle and connective tissue, or your ectoderm is actually your nervous tissue. So it's your, this is gonna be your central nervous system. This all turns into your spinal cord in your brain. Anyway, sorry. The ectoderm is also going to turn into your epidermis, and your mesoderm is going to turn into your dermis and your hypodermis. So, oh, sorry. So the nervous tissue, your brain and your spinal cord is just this little part right here. But the rest of your ectoderm will become your epidermis, to be your skin. All of this, the endoderm will be the lining of your gut. This will all be your muscles and your connective tissue, and your dermis and your hypodermis, right? Because those are made of connective tissue. So your dermis and hypodermis come from mesoderm, and your epidermis comes from ectoderm, which are just terms for the most basic differentiation that your cells undertake in the early embryo. So I thought that was pretty cool. From early embryo into adolescence, right? Once you get past this basic differentiation and then all the other differentiations that happen from there, right? So once this differentiates into all the different types of skin, thick skin, thin skin, the things that are in your skin, all of those different types of cells, the keratinocytes, the melanocytes, blah, blah, blah. Once this differentiates into all of your different types of muscle, all of your different types of connective tissue, and once this differentiates into all the different cell types that are lining the hollow organs of your body, and you're born, and you grow up and you become a young a tween, you know, or early teenager, and you hit puberty, your sebaceous glands, which were there, sitting there under your skin, dormant, become active and start secreting sebum, right? This is really sad. This was here in this PowerPoint before I like edited it. I just left it there, even though it's depressing. The effects of cumulative environmental assaults show after age 30. Like, that's really sad. Yeah, you start to like show fine lines and wrinkles and stuff after the age of 30. And you end up with more scaling and dermatitis. Lovely, isn't that wonderful? I should have put in the apocrine sweat glands are activated as well. I should just do that. And then finally, in old age, this is what we all have to look forward to. Epidermal replacement slows. So all of that mitotic activity in the stratum basale kind of slows down. So the skin that is sloughing off the top might actually exceed the rate of new cell production at the stratum basale in the epidermis. So your skin becomes thinner. And you also lose out on the keratinocytes and the waterproofness that it provides. So you lose more water through your skin, so you get dryness and then itchiness. You lose some of that subcutaneous fat, so the hypodermis thins, you lose that adipose tissue layer a little bit at least. And then the elasticity of that actually also decreases. So the elastic fibers tend to break down, leading to perpetual coldness. That won't be anything new to me. And wrinkles, naturally, right? Because that was kind of like puffing out your skin. And once that goes away, your skin starts to wrinkle a little bit. And then, of course, you have your increased risk of cancer because the melanocytes that were in the stratum basale of your epidermis, you'll have fewer of those as well, right? So the reproduction of all of your epidermal cells being produced in the stratum basale slow down, including melanocytes, which have the important function of shielding your cells against UV radiation, right? So you lose some of that protection. And then, of course, the dendritic cells that spread that melanin around, those also divide more slowly, and so you have fewer of those, etc. And yeah, that's about it. Any last questions before I guess I'll just stop the recording there. And then you guys, I'm gonna stay on, and you guys can ask any questions that you would like. Or if anybody's got a question right now, might as well just answer it and leave it on the recording. Anything? Or are you guys ready to take a break? At this point, we'd have a clean hour break if we just stop here. And then come back at 5:30 for a lecture. And lecture will just be a Q&A. So fully voluntary. The lecture that starts at 5:30, I will just, I'll just have this open and you can come in and ask me any questions that you have about the lab or the lecture. This will be our normal lecture hour. This will be in addition to the office hour that I'll have next week. So if you have questions about histology or the integument, I'll be happy to answer them during lecture tonight, starting at 5:30, or next week, Tuesday at 2 p.m., which I'll send you an email to remind you about as well. Does that sound okay to everybody?
Yeah.
Anybody still there? Oh, cool. There's still people here. Hi, guys.
Yes.
We got a chat. We got a chat. Let's see. What do we got? Yes. Thank you, Vanessa. Excellent. Okay, cool. All right, then, you guys. I'm going to sign off. Oops. What did I do? I didn't mean to do that, whatever it was. Whatever. I don't need to share. I'm over it. Okay, fine. I'll share then. Oh, fun with Zoom. Okay. All right, you guys, then I'm going to see if I can figure out how to stop the meeting, and then I will come back at 5:30 to start our meeting there, found in the same place. Everybody knows where to find our meeting, right?
Yes.
Okay, see you then. Have a nice little break, you guys. See you later, maybe.
You too, Professor.
Bye.
Bye.
Bye.